Welcome to the deep dive. Today we're tackling something really fundamental in conservation. How do we actually count animals in the wild reliably? It sounds basic, but it's often the hardest data to get. Traditional ways. Costly, difficult, takes ages. So we're doing a deep dive into a fascinating study that offers a potential solution. It cleverly mixes new tech with some, well, pretty old school biology. The paper's called "The Relationship Between eDNA Particle Concentration and Organism Abundance in Nature is Strengthened by Allometric Scaling," and the lead author is Dr. Matthew Yates.
Yeah, this is a really important piece of work. It gets right at the heart of a major hurdle for using environmental DNA, or eDNA, as a proper quantitative tool. We know eDNA is great for detecting if a species is somewhere, right? You find their DNA in the water, you know they're around.
Right, presence, absence, that's already useful.
Totally. But for things like, say, setting fishing quotas or figuring out if an endangered species is recovering, you need more. You need to know how many are there, abundance. And that's where it's been tricky. Lots of studies have compared eDNA concentration in water samples with actual population counts, but the connection, it's been okay, but not great.
What does okay but not great look like in numbers?
Well, the predictive power, the R-squared value, usually sits around 0.5 to maybe 0.6, something like 0.51, 0.57 in that ballpark.
So over 40% of the variation is just unexplained. That's a lot of uncertainty if you're trying to manage a population.
Exactly. It makes people hesitant to rely solely on eDNA for those big decisions. And this study, Yates and his team, they zeroed in on why that connection might be fuzzy. They proposed we've been thinking about eDNA production incorrectly.
How so? What was the assumption they challenged?
The sort of default thinking was that eDNA production scales kind of linearly with either the number of individuals or their total weight, their biomass.
Meaning like double the fish biomass, double the eDNA.
Pretty much, yeah. Or at least something close to that. But basic biology suggests that's probably wrong. It doesn't really work like that.
Okay, unpack that for us. Why is that linear assumption flawed? This feels like the core idea.
It boils down to something called allometric scaling. It sounds complicated, but it's just about how physiological rates change with body size. Think metabolism, breathing.
Or shedding skin and waste, which is where eDNA comes from.
Precisely. Think about, I don't know, a mouse versus an elephant. The mouse has a much higher metabolic rate per gram of its body weight. It lives faster, burns hotter, proportionally speaking.
Right, because of surface area to volume ratios and things like that?
Exactly. And that same principle likely applies to shedding DNA. A big fish per gram of its weight probably sheds fewer eDNA particles than the same weight made up of lots of tiny little fish.
Ah, okay. So if you have two lakes, both with 100 kilos of fish, but Lake A has 1,000 tiny fish, and Lake B has 5 huge ones.
Lake A is likely going to have way more eDNA floating around, even though the total biomass is identical.
That makes immediate sense. So just measuring total biomass misses a huge part of the picture. The size structure of the population, how the weight is distributed among individuals.
I got it. Metabolic processes, they don't scale linearly with mass, right? They scale according to a power function, usually something like mass to the power of 0.75 for metabolism.
And for surface area, which you'd think relates to shedding skin cells.
It's often a bit lower, maybe mass to the power of 0.6 or 0.65. The key thing is the exponent b in that equation, $1 goes idle MBB, is less than 1.
So if people were implicitly assuming BW1 by using biomass, wow, they were missing maybe 25–35% of the biological scaling effect right off the bat.
That's the potential scale of the issue, yes.
Yeah.
So the hypothesis becomes, if eDNA production is tied to these nonlinear processes, then eDNA itself should scale nonlinearly with mass, and if we account for that properly.
The correlation with abundance should get much, much better.
That's the prediction. Build the physics of biology into the model.
Okay, logical hypothesis. But testing it in the real world, across actual lakes, sounds like a massive undertaking. How did they set it up?
They picked a great model system, brook trout, Salvelinus fontinalis. Crucially, these fish show really big differences in average size between different populations.
And where do they study them?
9 different lakes across several Canadian national parks. Kootenay, Banff, Yoho, beautiful places. But yeah, requires serious field work.
I bet. Getting the ground truth abundance must have been the first hurdle. Mark recapture across 9 lakes.
Oh yeah, super intensive. They did full mark recapture surveys to get population counts for all fish bigger than 80 millimeters. But here's the critical part. They also meticulously measured and weighed samples of fish from each lake to get detailed size structure data.
That size data is key for their new metric, right?
Absolutely essential. Alongside catching fish, they collected water samples 1 liter each from 8 spots per lake, spread out near shore and in open water. Filtered them, ran qPCR to count DNA copies per liter, standard eDNA stuff there.
Okay, so they have the eDNA concentrations. What did they compare them against? The old ways first.
Yep. They tested 2 standard metrics. First, just individual density number of fish per hectare. Second, biomass density, total kilograms of fish per hectare. These are the kinds of things previous studies often used.
And those gave the meh results we talked about earlier.
We'll see in a second. But then the third metric was their novel one, allometrically scaled mass, or ASM.
How did they calculate that?
Okay, so for each population, they took the mass of each individual fish, raised it to a specific power. Initially, they used 0.73 based on known metabolic scaling for brook trout, summed that up across all fish, and then divided by the lake area.
So it's like biomass, but it gives proportionally less weight to the really big fish, accounting for that scaling effect.
Exactly. It weights the contribution of each fish based on this biological principle.
But using 0.73 from previous studies, isn't that still an assumption? What if brook trout in these lakes scale differently?
Good point. And I thought of that. While they started with 0.73 based on theory, they also designed the analysis to figure out what exponent best fit their actual eDNA data. They let the data speak.
Smart. So did the standard metrics perform as expected first? Confirm the baseline problem?
They did. When they correlated eDNA concentration with individual density, the R-squared was 0.59. With biomass density, it was a bit better, 0.63.
Okay, so right in that range we discussed. Decent, positive correlation, but lots of scatter.
Yep. Confirmed the status quo.
Hmm.
But then, the moment of truth, they ran the model using their new ASM metric.
Yeah.
The model fit shot up. The adjusted R-squared reached 0.78.
Wow. Okay, 0.63 to 0.78. That's a huge improvement. They basically cut the unexplained variation almost in half.
It's a massive jump in predictive power. And statistically, the ASM model was way better than the density model or the biomass model. The comparison statistics, the AIC values, showed clear separation.
So the hypothesis holds up beautifully. Accounting for how individual size affects shedding rates really is the key.
It seems to be. And remember your question about the assumed 0.73 exponent.
Yeah. Did the data agree with that starting point?
Get this. The exponent that the empirical data suggested was optimal, the one that best explained the eDNA patterns they observed, was 0.72.
Seriously, that's incredibly close to the 0.73 they picked based on metabolic theory.
Right. It's really strong evidence that this underlying physiological scaling is what's driving the relationship. The data screamed allometry matters.
That's pretty convincing. The credible interval they calculated, the likely range for the true exponent, was what?
It spanned from 0.47 up to 0.89. So while there's some uncertainty, the key takeaway is that the exponent is definitely significantly less than 1. The old linear assumption is just wrong. ASM or something like it is necessary.
Okay, huge implications here. One thing the study pointed out that really struck me was about how previous studies sometimes measured abundance itself. They often use proxies, right? Like catch per unit effort, CPUE.
Yeah, that's common. Easier to get than a full mark recapture census.
But in these 9 lakes, how well did CPUE actually correlate with the real abundance, or even with the eDNA?
Terribly. The correlation between CPUE and eDNA concentration was minuscule, an R-squared of just 0.1A. It barely correlated with the true density either.
So it makes you wonder, in those older studies showing weak eDNA correlations, how much of the noise was actually because the eDNA method was flawed versus how much was because the ground truth abundance measure, like CPUE, was itself unreliable?
That's a really critical point this study raises. Maybe eDNA was doing better than we thought, but it was being compared against poor benchmarks sometimes.
Okay, but the real promise here is using this predictively, right? For lakes where you don't have the expensive census data.
Exactly. And they showed that could work using a case study, Hidden Lake. It had undergone some fish removal, so they didn't have a current population estimate.
So they took eDNA samples from Hidden Lake.
Ran it through their validated ASM model, which gave them an estimate of the allometrically scaled mass per hectare for that lake.
But ASM isn't the final number managers need. They need fish per hectare, or kilograms per hectare. How do you get back to those?
You still need one more piece of information.
Yeah.
The size structure. You need to know, roughly, whether the ASM is made up of many small fish or fewer large fish.
And they didn't have that for Hidden Lake specifically.
Right. So they used a proxy. They took the size structure data from another nearby similar lake, Olive Lake, and assumed Hidden Lake was reasonably similar.
Okay, a practical workaround. And using that Olive Lake proxy structure,
They could convert the eDNA-derived ASM estimate for Hidden Lake back into traditional metrics. It predicted a population of over 3,200 individual trout.
that's incredibly useful. It means if you can get even a rough idea of the size structure, maybe from limited netting, historical data, or a nearby lake, you can potentially leverage eDNA for pretty good abundance estimates without a full census.
It's a pathway forward. And this isn't just for single-species qPCR studies either. The authors suggest this scaling principle has wider implications.
Like for metabarcoding, where you sequence all the DNA in a sample to see everything that's there.
Exactly. Metabarcoding often uses DNA read counts as a proxy for the biomass of different species. But if smaller organisms shed more DNA per gram,
Then your read counts might be naturally inflated for species dominated by smaller individuals, right? It could skew community biomass estimates unless you apply some kind of allometric correction there too.
precisely. The underlying mechanism how shedding scales with size is likely universal across many aquatic species.
Okay, this is clearly a breakthrough, but like any study, we need to talk limitations. What caveats did the researchers themselves point out?
Well, first, remember their mark recapture only counted fish 80 millimeters and up. They missed the really tiny age 0 fish.
And given the scaling principle, those tiny fish could be contributing disproportionately more eDNA per individual, right?
Potentially, yeah. So missing them might account for some of the variation the ASM model didn't explain. There's still that 22% unexplained variance.
What else? Environmental factors.
Big one. They deliberately didn't include environmental variables in this first model to isolate the effect of allometry. But they noted Temple Lake was much, much colder than the others, like 3.5 Celsius versus maybe 14 degrees on average elsewhere.
And its eDNA level.
Way lower than predicted by its ASM. Suggests temperature heavily influences either how much DNA the fish produce or how fast that DNA breaks down in the cold water. That needs to be layered in.
Makes sense. And you mentioned the physical lake shape earlier.
Yeah, the exact size and shape of the littoral zone, the shallow edges mattered. Changing assumptions about that actually shifted the best fit scaling exponent slightly. So having good bathymetry data, knowing the lake's physical structure, will also refine these models.
Okay, so limitations noted. Where does the research need to go next?
I think the most crucial next step is validating this at the individual level. This study correlated population averages. Now we need controlled lab or mesocosm experiments.
Like put fish of different known sizes in tanks and actually measure their individual eDNA shedding rates.
Exactly. To really pin down how shedding scales with mass and maybe even separate out DNA from slime versus waste, for example.
And testing it on more species, obviously.
Definitely. Brook trout are one thing, but scaling coefficients can vary a lot between different types of animals. We need to see how well this holds up for, say, carp or amphibians or invertebrates in different types of systems, rivers, oceans.
So species-specific and system-specific calibration will be needed.
Yes, which means there's still an upfront investment. To use this predictively, managers will likely need to do some initial intensive sampling in their specific system, get good abundance, and sign structure data to calibrate the eDNA model first.
But once calibrated.
Then the long-term monitoring could become much faster and cheaper, using just the eDNA samples and maybe occasional checks on the size structure.
So wrapping up, what's the core message you want listeners to take away from this deep dive?
I think it's that the biology of the organism itself, specifically how its size affects its basic physiological processes like shedding, is incredibly important for interpreting eDNA signals quantitatively. It's not just about the physics of DNA in water. It's about the ecology of the source. Incorporating this fundamental principle of allometric scaling seems absolutely key to unlocking the full potential of eDNA for estimating not just if animals are there, but truly how many. It elevates eDNA from just presence-absence to a real abundance tool.
And while getting that initial calibration data is still a hurdle, it's not easy work. The payoff is potentially huge. Getting reliable abundance estimates for fish in 9 remote lakes from just 8 water samples per lake plus some size data, I mean, that's a game changer compared to traditional methods. It really is an exciting prospect for anyone involved in monitoring wildlife or managing natural resources. So let's leave you with a final thought. The study found an optimal scaling exponent of 0.72, very close to the general metabolic scaling exponent of 0.75 that biologists often use. If 0.75 is already a pretty good first guess for many species, how many elusive or endangered animals, things we currently have almost no population data for, could we start getting abundance estimates for right now? Maybe all we need is their eDNA from a water sample and some basic size information that might already exist, perhaps tucked away in museum records or old field guides. Could we leverage that existing knowledge with this new approach to start filling in some major gaps in conservation? Something to think about.